Greetings, my name is Wesley Straub, and welcome to episode number 1 of Repligen's 2022 webinar series. Today's title, Custom Affinity Resin Development, right first time, Custom Affinity Resin Development with Unparalleled Diversity, introduces Repligen's ability to develop custom affinity resins in a really unique fashion. And it fits into Repligen's overall downstream portfolio that includes Opus and chromatography systems, because it enables users to reach some really important goals, which are number one, to develop processes faster, to execute them on a manufacturing scale with improved economics, and 3, really increase the capacity or productivity of a facility on a per square foot level. So to present this story today, we are really lucky to have Kevin Isett. He's a former co-founder of Avitide, which was acquired by Repligen in 2021. So he's been there since day one and can really tell the story in a really unique fashion. And then we have Kevin Chavarria. Kevin Chavarria recently joined Repligen and is going to walk us through some of the important aspects of quality and manufacturing and what the customer experience is like on the latter end of the process. All right, so I have 3 housekeeping items that I need to walk you through before we get started. First of all, this webinar is being recorded so that those who couldn't join us today can view it at a later date. Number 2, we are in a virtual environment still with COVID in this webinar, but we do like to have some interactivity. So at the bottom of your screen, you'll see a small button that says polls and Q&A. So if you click that, it'll open up a small screen to the side. We really like to understand a little bit about you. So if you answer the first poll question, we only have one at the beginning, that would be great. And then we also really encourage you to ask questions throughout the webinar. We do pull them together and we'll address them at the end in a Q&A with both Kevins. So feel free to ask questions throughout the webinar. And then third, well, that's it, right? So that'll do it. Already covered my Q&A bit. All right, so with that, I'm going to hand it off to Kevin Isett for today's webinar.
All right, thanks, Wes. Hi, everybody. Kevin Isett, former co-founder, CEO, and now the head of the Avitide business unit within Repligen. I like the title Unparalleled Ligand Diversity. It really underscores how we do things differently from the rest of the affinity chromatography development community. And I'll go into more of that later. But let's first start with a high-level view. Listen, to make a manufacturing process, it's typically broken down into 3 stages. There's the production, purification, formulation, fill, and finish. Avitide's sole focus is in the purification and advancing the technological state-of-the-art of purification technologies. So the current state-of-the-art typically employs ion exchange capture, followed by a series of polish steps. The drawback to that approach is each of those steps typically has an impact to yield, i.e. drug loss. So at each step, those drug losses accumulate to quite a large number, and that affects the amount of volumetric production you need to do in order to achieve your mass requirements. As noted by the use of Protein A, widely used in the industry for monoclonal antibodies, Avitide's aim is to standardize and to provide that same benefit, but agnostic of biopharmaceuticals. So why that matters, it really comes down to cost of goods and manufacturing economics. And said simply, yield, purification yield, drives manufacturing economics. So cost of goods can be simply thought as titer, how much you made in production, and yield, how much you lost in purification. The chart to the left shows a comparison between affinity-based capture purification processes and that of an ion exchange capture purification process. And what you see is a reduction in steps with affinity-based capture, again, because you have high yield and high purity in that capture step, it reduces the need for polishing steps subsequent to that. So that you see that subsequent reduction in steps as processing speed, but most profound to cost of goods is the increase in yield and thus the reduction in cost of goods, anywhere from 2 to 3x lower. Again, Protein A sets the standard in terms of cost-effective purification, And Avitide aims to provide that same performance to all biopharmaceuticals. As noted by our recent launch of 3 affinity chromatography resins towards AAV and gene therapy, Avipure AAV9, 8, and 2. We've also developed over 30 campaigns, again, developing affinity chromatography resins. So if we were to look at how we do this, it essentially is broken into 3 steps. So the first step is we discover ligands. The second step is we take those ligands and we develop affinity chromatography resins, and we prove the performance of those resins. We can do that in about 3 months' time, so each stage taking about 6 weeks. So 3 months, we can deliver from a start of a project, a prototype affinity resin. We can then transfer that prototype affinity resin into our quality and manufacturing environment, scale that up, and supply that resin for use in GMP manufacturing in 5 months' time. This is a marked increase in terms of service timelines when compared to other technologies in this space. So you're probably wondering, how can you deliver it so fast, and what's the underlying technology? As I alluded to in the first slide or in the intro slide, it really comes down to an incredible diversity of affinity ligands. When we launched Avitide in 2013, we had 9 different affinity ligand scaffolds. Today, we have well over 50 novel and unique affinity ligand scaffolds, all protein-based, all stable in sodium hydroxide, and designed for the rigors of bioprocess purification. Again, we do not employ animal immunization. We are an in vitro, empirically driven discovery platform, and we can discover these ligands within 3 weeks' time. So what we've learned from this increase in diversity and having multiple scaffolds is that there is not one scaffold that works for all applications and all molecules. So by having a toolkit approach, we always find the scaffolds that work for each particular application. And again, that's proven to be an absolute requirement to deliver for all the different campaigns that we've delivered on. So after we take it out of ligand discovery and into resin development, we customarily prove the performance of our resins with our partners' bioprocess feed streams. And this is of critical importance. So not only do we deliver a solution that we've proven meets your specifications in 3 months' time, we actually tech transfer that information and those protocols and methods to our customers, along with the resins. And because we're a part of the greater Repligen organization now, we can offer those in a variety of pre-pack OPUS formats. So this slide is a summary of all the projects that we've done and delivered for, just to give you a sense of the types of molecules and what stage of development those molecules are in. So in terms of the program stage, we can enter a partnership with a customer and develop a chromatography solution anywhere from preclinical to a commercial program. Drug modalities, very diverse. Recombinant proteins, growth factors, hormones, enzymes, to viral vectors, to vaccines, to antibody-like molecules such as Fc fusion proteins, antibody fragments, bispecific antibodies, and some of the more novel binding scaffolds. To vaccines, I think which I just said, as well as enveloped particles such as exosomes. The project scope can vary. We sort of see 3 types. There's a single resin, which is targeted towards a single molecule, and that can just give high yield, high purity, and a relatively straightforward capture. The second is around developing a platform affinity resin that can be used, that targets a conserved motif that can be used across multiple family of proteins or family of molecules. So that particular customer can use one resin across a number of different programs. And the third is around high resolution separations, and quite frankly, where Avitide is a pioneer. Here, we're not only just binding to the molecule, but we're binding to a very specific epitope on the molecule that discriminates between what is the drug substance to what is a closely related product impurity. And the power of that is we know by what we're capturing, it is the drug substance, and all those product impurities are in the flow through. So that can be a very powerful tool, especially when drug product quality around these impurities is an absolute mandate. And again, across these programs, Avitide has always been able to discover ligands, and we've always been able to deliver resins. So getting into some of the technical elements as around each of these chromatography projects, each of these three graphs somewhat summarizes how our customers judge our ability to develop chromatography resins. It goes purity. So these are essentially, each of these dots is a unique custom affinity resin delivery. And so what we have is the chart on the left is based on purity. So some of our customers ask us to optimize the wash. Some customers do not. Either way, we're fine with that, and we'll take your guidance. But minimally, we deliver affinity chromatography resins that have greater than 2 log host cell impurity reduction. And that's across mammalian and microbial hosts, intracellular, as well as secreted expression. The center graph is around caustic stability or sodium hydroxide stability and proving column lifetime. So again, Avitide will do this work as a part of the project and will prove that you can actually extend the column lifetime before we deliver it to you. So this is essentially a parity plot where on the x-axis is the specification that is requested of us by our customer and what we delivered from that campaign. And again, each of these blue dots is a unique custom affinity resin delivery. So as you can see, we are either at or above consistently showing stable and long column lifetime. And this is in 0.1 molar sodium hydroxide. We've also been able to develop and deliver resins that can withstand 0.5 molar sodium hydroxide. Most notable are our recent launch of our Avipure AEV resins, which can withstand greater than 20 cycles in 0.5 molar sodium hydroxide. The third is around binding capacity. So here again is a parity plot on the x-axis is what our customers have asked us to develop in terms of capacity. And on the y-axis is what we've delivered from each of these projects. So in the main, we're not restricted to lower capacities with our ligands and how we optimize the chemistry, ligand density, ligand orientation, and the base speed. We can dial that in for many different types of molecules, whether they be sub 10 kilodalton recombinant proteins, all the way up to very large, greater than 500 kilodalton recombinant proteins to some of the more novel viral vectors. And again, in the main, we've been able to consistently deliver high binding capacities or relevant binding capacities to a wide variety of these projects. Noted in the asterisk in the arrow, some of our customers, we may present them an option. You know, there may be a resin that has exquisite purity, but slightly lower capacity. And in this example, that customer chose that resin. Again, so giving our customers option, allowing them to decide what is optimal for their process and facility fit, and then they can take those resins forward. So with that, I'm going to hand it over to Kevin Chavarria, who will walk through ligand manufacturing and some of the other offerings that Avitide and Repligen have to offer our customers.
Thanks, Kevin. Hi, everyone. I'm Kevin Chavarria. I'm the Director of Business Development for Avitide, and I'm going to talk a little bit about Avitide's manufacturing, support, and supply capabilities, and really focus on that post-development relationship that make our partners successful. Repligen acquired Avitide in September of 2021, and there were just a ton of synergies. It was really the perfect fit. At the top of the list, Repligen is already the largest affinity ligand manufacturer in the world, supplying over 80% of the entire protein A ligand market. With that, Avitide custom and catalog resins are able to benefit from that robust global manufacturing footprint, especially the redundancy in global manufacturing sites. As you can see here, Repligen also plans to add additional capacity in manufacturing in Hopkinton, Massachusetts. Diving into the manufacturing, Repligen has 18 global manufacturing sites, 3 of which are ISO 9001 sites dedicated to resin coupling, and there are additional 3 sites dedicated to ligand production. As we've learned over the last few years with COVID, it's especially essential to have a secure supply chain. Repligen affords us just that, a secure ligand and raw material supply chain, as well as safety stock programs for customer finished goods. It's also important to note that that same extensive regulatory support package that comes with our catalog Avipure products also comes with our custom resins. We also include residual ligand support analytics with every single project. Support-wise, since custom projects are completely exclusive to that customer, there's also a valuable flow of intellectual property to our partners from our SMEs. Finally, our custom resins can be delivered in pre-pack Opus columns, so there's a significant reduction in lead time since everything is now in-house. This slide is a great depiction of our entire affinity chromatography integrated solution. As you already know, we can develop and manufacture a custom resin in a speed in which you've never seen before to your specs. We can then deliver this resin in pre-packed Opus columns ranging from the PD scale all the way through 80 centimeter columns, meaning you do not need packing or unpacking hardware or the validation that comes with that. Finally, you can run your capture step with our single-use artisan chromatography system, which has been strategically engineered to address traditional single-use flow path pain points. The best-in-class fluid dynamics produce high yield and high resolution solutions. Our end-to-end solution ultimately allows you to move at a rapid pace, drastically improve manufacturing economics, and potentially support more programs. Thank you everyone for your time, and now I'll hand it off to Wes for Q&A.
Okay, thanks, Kevin. Kevin C, we have 5 questions today from 5 different people. I should mention that to the audience, if you'd like to submit a question, you can still do so. And if you like a question, sometimes we run out of times. You can use the upvote function to basically push its priority up and let us know that that question really should be answered in the time that we have here today, right? If we don't answer your question today, we send you an email offline and answer that later. All right, so I'm going to start our questions. Actually, we have one from Hans that's more on the development side of things, so I think it's a good starting point. And Hans asks, how many ligands can be brought forward in parallel? Kevin, I think that one might be a great question for you since you were there in development of the platform.
Sure, yeah. So we, again, interrogate very large libraries, mainly in excess of 1 to the 12th across 50 plus scaffolds. That quickly, again, our job within that first 3 weeks is to eliminate diversity that isn't applicable. So we can drive that down to the thousands, tens of thousands of ligands, ultimately at the end of the 6 weeks, getting down to, tens of ligands that demonstrate biophysical properties, affinity, selectivity, sodium hydroxide stability, that meets our partners' selectivity requirements.
Okay. And then again, maybe we're going to go to the second question that's also kind of on the development side of things, specifically around the affinity ligand, right? DT asks how to estimate stability of the affinity ligand by analyzing leaching of the affinity ligand in the final product. So is the stability measured, I guess I would say, is stability measured before it's coupled to the resin, or is it done as well afterwards, or both?
Yeah, so typically we can get an early indication as to first the ligand. Are there intricacies, or is there something biophysically about that ligand that is unstable, particularly in caustic conditions? And again, we can do that and dial into any particular flaws of that ligand, either for re-engineering or to avoid that ligand and move forward with other more promising candidates. Second to that is, again, we prove the performance of the affinity resins we develop with our partners' feed streams. And so part of that is also cycling the resin and either imitating or replicating column reuse conditions, again, in sodium hydroxide. So if we see a diminishing effect in terms of performance, that's an indication. And then lastly, when we support these products for manufacture and supply for use in GMP manufacturing, that comes with residual ligand analytics, which again, you can quantify the actual amount of any residual ligand leachate.
Okay. Again, we're going to stay on that development side of things. So we're going to go out of sequence here, but John asks, what's the volume of resin available after the 3-month development process, to kind of probably give an idea of how much material they have to work with at that point in the process?
Sure. Yeah, we can typically be flexible, but common is around 10 mils. That can be slurry or pre-packed through in a variety of different sort of Opus formats. That's very typical. Usually after that 10 mil delivery, probably really quick out thereafter, our customers ask for lab scale quantities, and we can deliver up to a liter within about a 4 to 6 week time frame. So again, they can do process development work and things of that nature before we formally bring that resin into our quality management and supply and manufacturing environment.
Okay. So about 10 mils in the beginning. All right, so we have another question that kind of takes us back a little bit earlier in development. But what are the and it's a good question, I think, because the scaffolds play the diversity of scaffolds play such a big role in the platform. What are the typical molecular weight of the Avitide ligands? But I'll take that as a scaffold.
Yeah, and they range in a variety of different sizes. Almost equally important to the size is also the conformation, the shape, how they interact with different sort of drug surface features. So anywhere from tens of amino acids to about 5, 6, 7 kilodaltons, 10 kilodaltons max.
Yeah. Okay. Okay. Then we have two questions. Maybe we can do both at the same time, right? Around the support of it. The first one is the affinity resin based on agarose. If not, what is the resin backbone? But we have a very similar question, which takes it off to different supports, which is, can the Avitide ligands be applied to membranes? I think maybe we talk about residence time around the bead and tackle both those at the same time.
Sure. So specific question around what supports we use, Wes?
Yeah. How about the first one? The first one is what bead do we use? Is it based on agarose?
Yeah, it's based on hardened agarose, again, sourced. You can probably look at the commercial products that are available now, which we source from Purolite. In terms of, are the ligands amenable to membranes? We don't have any membrane products currently, but we have demonstrated that technically in the past. But we were also seeing faster flow rates too with the agarose-based resins, most notable with Avipure AAV resins, which we can flow at a 1 minute residence time and still achieve very high bonding capacities.
Yeah. Yes, that's the question, right? So I think if around the membrane, I mean, if you're using a membrane, you really want to get the residence. Clearly, you want to get the residence time down if you're interested in a membrane. But I think hopefully maybe the 1 minute would suffice on the agarose bead right now. All right, so you did mention, so we are going to have a webinar specifically on the Avipure AAV 289 resins next month, but someone did hear you mention it. So Ben asked, does Avipure just work for serotypes 2, 8, and 9?
Say it again, I'm sorry, Wes, I missed that.
Oh yeah, Ben asked. So Ben heard you mention today's topic was really focused on the custom resin development platform. But Ben heard you mention on that we do have available AAV serotype 289 specific resins, and Ben's asking if we have resins outside of those 3 serotypes.
We are in various stages of development. So our goal is to address the major serotypes that are being used today in manufacturing and gene therapy for AAV, which is 9, 8, and 2. We do have development of other serotypes, and moreover, the world is moving towards more engineered capsids. So we can deliver a custom resin to those engineered, highly specific capsids in about 2.5 to 3 months' time. So if that's of interest, then that would definitely be a customized resin that we could work with that particular customer on.
Okay. So I guess maybe the take home, like if you do have a serotype or an engineered serotype, an engineered capsid that you're interested in, it might not be covered in the 289, but definitely worth a conversation.
Yeah, absolutely.
Okay. So we do have a question from Caleb. Do different ligands, it's probably around if you do different projects. I think Caleb's really getting at like if you do several different projects or you bring multiple ligands forward in parallel, do different ligands impact the resin pressure flow curves?
Short answer is no. We haven't seen that for over 30 campaigns. And again, we're looking at different ligands, different scaffold types, different ligand densities even. We have never observed pressure flow issues.
Okay. All right, good. And then I might turn over to Kevin Chavarria as we kind of I tried to answer bundle all the development questions into one. So Kevin Chavarria, like more like once the resins developed are basically both during development and manufacturing on the GMP, can the resin be packed in OPUS columns at both the development and GMP stages? And I think maybe in connection with that is maybe what sizes, like can you get all sizes at both stages?
Yeah, so the short answer is yes. The stages are from 5 microliters all the way through 80 centimeter columns, and that significantly reduces the lead time because you're getting everything from us in-house.
Okay. So on that one though, right? So that's a good question that you mentioned, like the resin in-house. Like maybe you could like talk a little bit about the advantages of having both the resin developed in-house and what it's like to transfer that over to an OPUS pre-pack column.
Yeah, so since Avitide has been acquired by Repligen, everything comes from our teams. So we work directly with the BSSSs that cover the OPUS columns. So once your resin's developed, we work with them directly to find out how quickly you need it, and we can really coordinate all that in-house.
Okay. All right. So Daniel asks, we have a really good one. Can you give more details on how ligand scaffold libraries are screened? I don't know how much detail we can go in there, but Kevin, I said maybe we can talk about what that screening process is like and how we ensure that we get to a ligand that meets the specifications of the customer.
Sure. Yeah. So, I mean, we don't employ animal immunization approaches for a number of reasons. We mainly do because what is immunogenic, it may not be important for bioseparation. Those two are a strong correlation. The second part of that is we don't employ in silico analyses at all, because no matter what the model predicts, you have to make it and empirically prove that it does what the model says it ought to do, right? Everything we do is empirically driven. So it's a protein engineering platform. We employ a number of discovery selection-based technologies to quickly and empirically drill down to what ligands give us the selectivity, the affinity, the stability that our partners need for that particular application. And again, very high throughput. So if anyone were to ever visit our site, which a number of our customers have, we can walk you through our workflows, and you can see at firsthand the level of automation and high throughput workflows that enable us to do what we do so quickly and efficiently.
All right. We have one from Anthony, which is I don't know which one of you two want to take this one, but are all of the ligand scaffolds suitable for regulatory approval as a leach contaminant? And we have one coupled to that we'll get to next.
Yes, the short answer is yes. We wouldn't employ a scaffold if it wasn't able to meet regulatory scrutiny. And again, we would support our custom resins exactly the same as we support our catalog products that are currently available.
Yeah. All right. So we do have a couple more questions in the background. We will answer those. We're at 11:30, so we're up on time. I'm gonna just because it's so related to the ligand again and the regulatory side of the thing. Maybe Kevin Chavarria, maybe you can answer. Do you provide a detection tools, ELISA, for measuring ligand leaching? And I would say specifically, we do that, and we announced that for both the AAV289, but do we do that for the custom?
Yeah, I can take that one. The same way.
Yeah.
Go ahead, Kevin. Sorry.
No, we do provide residual ligand analytics in the form of ELISA kits. However, we've also been asked on custom resins. Some of our customers are migrating to characterizing their host cell impurities and the residual ligands using mass spec methods, and we can also support those efforts as well.
Okay. All right. So with that, we wrap up episode number 1 of our 2022 webinar series. Both Kevins, thank you for joining us today, presenting and answering all the questions that came through. If your question was not answered during this Q&A session, then again, we will follow up with you within 24 hours with an answer by email. And with that, I hope to see you next month for episode number 2, which is around affinity resins, but specifically around the AAV 289 affinity resins that are available on a catalog level. Thank you and take care.